Thank you for joining us today for this CAST webinar. I'm Kent Chesky, and I serve as the Executive Vice President for the Council for Agricultural Science and Technology, or CAST. Very pleased to share with you our most recent CAST issue paper, Food Biofortification, Reaping the Benefits of Science to Overcome Hidden Hunger. As noted on the agenda, we will have a question and answer session with a panel of a group of our task force authors following the main presentation. And we'll open up the chat box in just a few minutes for you to submit any questions in advance. Next slide. CAST was formed in 1972 to provide unbiased, credible, science-based information about food and agriculture to policymakers, the media, private sector, and the public. CAST is a membership-based organization comprised of scientific and professional societies, nonprofit organizations, universities, companies, and many individuals. It is through our network of members and supporters that we select our CAST topics for CAST papers and access volunteer scientists, economists, engineers, and other subject matter experts who help us produce our papers. Over the last 13 years, over 1,500 professionals have graciously volunteered their time, energy, and expertise to serve as authors of our papers and reviewers who ensure that each is unbiased and based upon sound science. CAST produces several types of publications. Accompanying each publication is a one-page summary called an Ag Quickcast. And we also started producing student study guides for use by educators and students. The use of social media is an important part of our science communication efforts, and I invite you to join us on any of these platforms. Today's webinar is being recorded and will be available for viewing later on our website. Toward the end of our main presentation, we'll post a message that the chat box is open and you can start submitting your questions. The task force chair for this paper was Dr. Howarth Bouis, International Food Policy Research Institute. Dr. Bouis is a recipient of the 2016 World Food Prize, pioneering the concept of biofortification in the early 1990s, and founded HarvestPlus in 2003. He's devoted his career into turning this idea into a global movement that reaches more than 50 million people in smallholder farming families in low-income countries. After stepping down as the HarvestPlus CEO in July of 2019, he continues to actively champion biofortification and the work of HarvestPlus in the international agriculture and nutrition communities. I'd like to now turn the program over to Dr. Bouis.
Ken, thank you for that introduction, and thank you to CAST for the opportunity to publish this paper about biofortification and to present the findings here. This is a list of the task force members. I won't read out all their names, but I'd like to especially thank Michael Lipton and Hans de Steur for their careful reviews of the paper. Before I introduce the speakers, let me talk a little bit about how biofortification got started for me. As I was attending nutrition conferences in the 1990s, there was a great deal of interest in supplementation and fortification interventions to solve the problem of mineral and vitamin deficiencies. And from my perspective, they had two fundamental problems. The first is that they have recurrent annual costs. So for example, over the past 20 years, 10 billion vitamin A supplements have been given out at a minimum cost of $1 per supplement. And the supplementation and fortification do not address the underlying problem, which is poor quality diets. So you have to keep intervening in the same way at the same cost year after year. But it struck me that modern varieties were being consumed 3 times a day all over South Asia. For example, what if we had made those modern varieties in the very beginning high iron and high zinc? Then maybe these problems of iron deficiency and zinc deficiency wouldn't be present. So the question was, can we get the plants to do the work? So it was a nice idea, but I really wasn't sure whether it would work or not. And I sort of identified 4 fundamental questions that had to be answered. And you had to get positive answers to each one of these questions. And if you did get 4 positive answers, then biofortification could work. So the first question was the cost effectiveness. biofortification would have to be lower cost than existing interventions, else it wouldn't be worthwhile to implement biofortification. The second was, if you did get the plants to do the work and you did put more nutrients in the plants, would the extra nutrients be enough and be sufficiently bioavailable to improve mineral and vitamin status? The third question was, can you combine the nutrient densities with high yields? If there was going to be a trade-off in yields between better nutrient content, but you had lower yields, and farmers would never adopt the varieties. So you had to combine high yields and high nutrient densities. And then the fourth question was whether farmers would adopt the varieties and whether consumers would accept the varieties. So with those four fundamental questions in mind, Matin Qaim from the University of Goettingen will talk about the cost effectiveness of biofortification. Saurabh Mehta from Cornell University will talk about the nutrition studies. Torbert Rocheford from Purdue University will talk about the plant breeding activities. Jan Lowe and my fellow laureate will talk about scaling up delivery strategies for individual crops. Ekin Birol from HarvestPlus will talk about getting institutions all along the value chain to mainstream biofortification in their everyday activities. Dominique van der Straeten from Ghent University will talk about how transgenic methods can add multiple nutrients, multiple traits into varieties much more efficiently and much more easily than conventional breeding, and so can greatly increase the impact of biofortification. Then I'll come back at the end to talk about our policy conclusions and the way forward. Thank you.
Okay, so Howdy Bouis has already introduced the idea of biofortification, so let me directly jump into the question, why is it needed? Well, most of you are aware of the fact that micronutrient deficiencies are a widespread problem. More than 2 billion people are affected by deficiencies in different minerals and vitamins, and this is leading to really severe health problems. Poor people are particularly affected by these problems, and the reason is simple. Poor people, first of all, try to invest the little money they have in cheap calories, so their diets are dominated by staple foods, which are good sources of calories, but are usually low in terms of micronutrient density. The long-run goal should be to have everyone in a situation to have a sufficient income and good knowledge to afford a diverse diet with lots of fruits and vegetables. But we do know that in the short and medium run, this cannot be achieved. There are problems of access, seasonality, affordability, and therefore low-cost sustainable micronutrient interventions are required. Supplementation and industrial fortification are two commonly used micronutrient interventions that have been established quite a while ago. They are successful, but one of the limitations of these approaches is that they have relatively high recurrent costs. And this also means that their coverage, and especially their coverage in rural areas, is often limited. And this is one of the problems that biofortification wants to address. Here are a couple of examples of biofortified crops that have already been developed and released. Those have been developed with conventional breeding approaches. There are also examples where transgenic breeding approaches have been used. You'll hear more about this in later presentations. I want to focus now on a question that Howdy has raised, namely the question, is it actually cost effective to invest into the breeding of biofortified crops? And this graph here is based on a systematic review of all the cost effectiveness studies for different micronutrient interventions that have evaluated cost effectiveness in terms of the cost per DALY saved, and DALY stands for disability adjusted life year. So how much does it cost with a particular intervention to save one healthy life year? All of these are different studies, and the first thing that we see is most of these different micronutrient interventions are highly cost effective. They stay below the World Bank threshold for high cost effectiveness of around $270 per DALI saved. The second thing that's interesting to observe is that among the lowest cost interventions are biofortified crop examples. And the reason is that they don't have recurrent costs, or hardly any recurrent costs at least, because once a biofortified crop variety has been developed, the same technology can be used in many different situations, and many can benefit year after year. Farmers can reproduce the seeds at no recurrent costs. The third important thing to realize here is that yes, but there are also biofortified crops examples where the cost per DALI saved is somewhat higher. And this means there's no automatism. I mean, if you're targeting a crop that's not widely consumed in a particular context, or you're unsuccessful to get sufficiently high micronutrient contents into the crop, or there are any access or acceptance problem, there may certainly then be a lower cost effectiveness. And these are things that the coming presenters will address in more detail. Thank you.
Hello everyone. Thank you for this opportunity to share a brief synthesis of the evidence generated so far in support of agronomically biofortified crops. Overall, 17 bioavailability and 15 efficacy trials have been conducted, largely focused on iron, vitamin A, and zinc biofortified crops. In the interest of time, I've only included some of the highlights on this slide. For example, for iron, the target crops have included beans, rice, and pearl millet, and efficacy trials have mostly been conducted in India, Philippines, Mexico, and Rwanda among adult and adolescent women, school-age children, and also school-age children. 2 meta-analyses have shown that biofortified crops are efficacious in improving iron status, as reflected by ferritin concentrations and total body iron, particularly in those who were deficient at baseline. Similarly, efficacy studies have also shown that certain cognitive and physical fitness-related outcomes are improved as a result of intake of biofortified crops. Many more studies have been conducted with vitamin A biofortified crops in settings ranging from Bangladesh, Mozambique, South Africa, Uganda, USA, Zimbabwe, and Zambia. Target crops have included maize, cassava, and sweet potato, and populations that have been studied include adults as well as children. Key findings have included increased absorption of provitamin A carotenoids and also increase in storage biomarkers. Some studies have also reported functional outcomes such as increase in pupillary responsiveness in those with marginal vitamin A status and decrease in duration of diarrhea. So what's left on the research agenda at this point? We of course need better biomarkers, and that's a challenge across the entire nutrition community. Better intake sensitive biomarkers, better predictive biomarkers, they are both needed, and HarvestPlus is invested in some of these opportunities as well, and particularly in the area of zinc biomarkers. Parallel efforts by HarvestPlus are also focusing on increasing bioavailability further. Some of the work in the pipeline also is focused on adding more micronutrients and also looking at prebiotics in crops and their effects on human health, as well as evaluating the efficacy of multiple biofortified crops, particularly in even younger populations such as infants or in the first 1,000 days of life. Thank you.
Hi, I'm Torbert Rocheford from Purdue University, and I'm going to talk to you about plant breeding. Plant breeding is a long-term, ongoing process. It's been going on for thousands of years. As long as you have plants and people in the same place, plant breeding happens. And so it involves the interplay of people, science, plants, but also it's not limited to the plants and science and people. Culture, food, and society have a large impact on plant breeding because you can breed something, and if nobody likes it, doesn't use it, doesn't eat it, it's not too successful. So it's a very dynamic endeavor. And science has had an impact on plant breeding since about the early 1900s. Before it was largely visual selection by humans. And breeding for micronutrients has only been ongoing for about 15 years. And breeding takes time to make progress. So there's considerable upside potential because it's only a recent effort to breed for micronutrients in a systematic, scientific, coordinated way. And so new technologies also will accelerate progress. This is genomic technologies and analytical technologies that enable faster, cheaper, quicker measurements of the micronutrients and empower more rapid selection programs. The outlook for plant breeding continues to be very positive because of these. And for example, the genetics underlying these traits is not as complicated as traits such as grain yield. So the new technologies in genomics may, in particular, help select for micronutrients. And what you see here is an overview of a lot of the efforts coordinated by HarvestPlus. And there's been a lot of varieties released and being tested in over 30 countries. And already they're providing daily nutrition requirements of up to 80% for iron. And we're using new varieties from HarvestPlus in pearl millet or in dry beans. And for zinc, you have wheat at 50%, rice at 40%, and maize at 70%, providing the levels of that percentage of the needed daily zinc. And vitamin A is another success story, with sweet potato and cassava providing 100%, if you eat the appropriate amounts. And maize is now 50%, which is much improved from where it was not too long ago. So there's been good progress, and the technologies and gains will not only enable higher levels of these micronutrients, but will also mean we can develop varieties for other countries so more people will benefit.
Lots of biofortified varieties have been developed, but if they don't get out to those who need them, then that effort is truly a waste. I'm Jan Lowe from the International Potato Center, and I want to highlight key strategies and lessons learned in disseminating biofortified crops. Different models to deliver biofortified crops have been tested, some of these at large scale, permitting a good understanding of how cost-effective they are. Several models focus exclusively on improving nutritional outcomes, particularly of young children and pregnant and lactating women. To reach these most at-risk groups, integrating with a strong nutrition program to improve overall dietary practice is most desirable. Another focus has been on improving food security and incomes. This requires getting biofortified varieties well integrated into the value chain, from seed through consumption. Critical to success is having a highly acceptable, well-performing biofortified variety. To ensure this, farmers, traders, and consumers need to be involved in the variety selection process. Otherwise, the risk of ultimate rejection is high. But the greatest challenge has been tackling what we call the seed system bottleneck. Particularly in Africa, weak existing seed systems are the norm. Moreover, there are distinct kinds of planting material that drive how seed can be delivered. Vegetatively propagated crops like sweet potato and cassava, and self-pollinated crops like beans, wheat, and rice are dominated by informal seed systems. Farmers can often retain their own planting material for years and commonly share seed with other farmers. Because of this, private sector seed companies avoid marketing these seeds. Over time, diseases accumulate in the recycled seed and yields decline significantly. Selected farmers, however, can be and have been trained as quality seed, business-oriented multipliers for their communities. HarvestPlus and their partners have successfully implemented a model in which households receiving biofortified seed for free pass seed on to 3 other families in the following season, the so-called payback system. Hybrid varieties of vitamin A maize and iron pearl millet are at the other end of the spectrum. One must purchase seed annually to sustain high yields of hybrids. However, we have learned that most commercial seed companies need support to manage the initial risk of taking on a new variety. For instance, in Zambia, support for small packets of vitamin A maize was provided to enable farmers to get to know these varieties at low cost. Over time, farmers became willing to pay for the more standard size 50-kilogram bag of seed. We know that willingness to pay for seed is driven by consumers' demand for the final product. We have learned which varieties to use from diverse types of studies evaluating consumer acceptance of and willingness to pay for biofortified varieties. We have found that including nutrition information about the variety increases demand. We have experienced the positive impact of promotion campaigns in building awareness, such as a video with pop stars promoting iron beans in Rwanda, and having trusted members of society advocate for eating nutritious foods. Moving forward, expanding public-private partnerships will continue to be key, but we must support the growing awareness among consumers worldwide of the critical importance of good nutrition, and integrate biofortified crops into more multi-crop dissemination programs.
My colleagues have just presented the evidence on the feasibility of breeding for higher micronutrient content, and doing so without giving up on yield and climate resilience, on the cost-effectiveness and efficacy of biofortification in tackling micronutrient deficiencies and in improving health outcomes, and on the acceptability of biofortified crops and foods by farmers and consumers alike. Given this evidence base and the ever-increasing need for affordable, nutritious food for all, biofortified staple crops are being scaled to become the new normal. As HarvestPlus, we're implementing a 5-pronged strategy to catalyze sustainable scaling of biofortified staple crops and foods. These prongs are: 1. Mainstreaming of micronutrient traits in public breeding programs at international and national levels and in the private sector breeding programs. 2. Embedding of biofortification in national and regional policies and regulations, and especially in public programs. 3. Integration of biofortification in international financial institutions, agricultural, health, and multisectoral loans. 4. Inclusion of biofortified crops and foods in humanitarian programs, the need for which is unfortunately growing due to the pandemic, conflict, and climate crisis. And 5. Enabling and facilitating of enterprises of all sizes operating along the staple crop value chains to make the switch from non-biofortified to biofortified for seed, for grain, for ingredients, and for food. There has been some great movement in the right direction in the past few years. For example, 24 countries have now included biofortification in their national agricultural and/or nutrition agendas, strategies, and policies. And we're now also seeing inclusion of biofortified crops and foods in public programs, such as school feeding programs, input subsidy programs, and food distribution programs. Given several international financial institutions' commitment to improved nutrition outcomes, many of them, such as the World Bank, African Development Bank, and IFAD, are including biofortification in their loans. UN agencies such as the FAO and UNICEF are recommending biofortified crops and foods as a cost-effective opportunity to improve diets. And several humanitarian organizations, most notably the World Food Programme, are including biofortified planting material and food in their procurement policies and relief packages. On the research side, the International Agricultural Research Centres, as well as several national agricultural research systems, such as those in Brazil, China, and India, are mainstreaming nutrient breeding in their breeding programs. At the same time, several food companies of all sizes are experimenting and working with biofortified ingredients to produce healthier processed foods. These developments are especially impressive since biofortification is still a young technology. Given the sound evidence base and strong buy-in from all stakeholders, we expect biofortification to go viral in the next decade and to bring us closer to reaching SDG 2. Thank you.
I will briefly illustrate how the application of genetic engineering can boost the realization of multi-biofortified crops. For those of you who are unfamiliar with gene engineering, I will present the concept in a nutshell. The most commonly used method is a variant of a natural system of plant transformation based on Agrobacterium tumefaciens. Agrobacterium is a soil bacterium which infects plants, causing a plant cancer called a crown gall. In that process, the bacterium transfers a piece of DNA bordered by short repeated elements called left and right border sequences, shown as the red squares here below, to the plant, hence inserting a few bacterial genes in the plant genome. Plant gene engineering copies this natural system, putting a gene or genes of interest with their controlling elements between left and right border sequences, allowing Agrobacterium to transfer the entire transgene cassette to the plant genome. It's important to emphasize that this is a natural process and that several crop species, including sweet potato, banana and yams, were recently shown to have been naturally transformed by Agrobacterium in the past. As for any technology, genetic engineering has its pros and cons. First, it offers the ability to control tissue specificity, for example, the accumulation of provitamin A in rice kernels, as was done for golden rice. Second, results can be obtained in a much shorter time frame as compared to breeding, which requires many more generations. Third, gene engineering allows transfer of well-defined genes only, as opposed to the potential transfer of multiple untargeted genes upon breeding. Breeding, of course, also has its advantages as compared to gene engineering, and we can come back to that in the discussion if you wish. The way to move forward in reducing hidden hunger is to combine several vitamin and mineral traits together with agronomic traits in staple crops, and thus render these more beneficial both for farmers and for consumers. Now, that goal can be reached much faster by embracing gene engineering and combine it with breeding technologies. For example, one can introduce the genes related to 2 different vitamin and 2 different mineral pathways, all together with a gene conferring an interesting agronomic trait, for instance, reduced water use, on a single DNA cassette, and then transfer this cassette to the genome of different crop plants using Agrobacterium technology. In addition, one can exploit conventionally bred varieties, which for instance would already have a higher content in a certain third mineral or third vitamin, as a background to transfer the DNA cassette. In addition, with the advent of novel breeding technologies based on CRISPR-Cas, it's possible to obtain a targeted insertion in a safe harbor location in the plant genome. In conclusion, I'm advocating a rational combination of different technologies, but this should go hand in hand with nutrition education and dietary diversification. Thank you for your attention.
Thank you to all the speakers. I think ample evidence has been presented to show positive answers to the 4 fundamental questions that we posed at the beginning. The plant breeding has been successful. The nutrition studies have shown even improved functional outcomes with the levels of nutrients that have been bred. They've been bred into high-yielding backgrounds. They've been adopted by farmers and consumers. So we've also been able to show the delivery, get a lot of institutions to start mainstreaming biofortification, and it's highly cost effective. So one extra thing that I'll point out, everyone's focused on COVID. There are certain advantages of biofortified crops under COVID because incomes are going down, consumption of non-staple foods is going down, dietary quality is getting worse, but the poor protect their consumption of food staples to keep from going hungry, despite the decline in incomes. So one way that the poor can improve their mineral and vitamin intakes is to substitute biofortified crops for non-biofortified at no extra cost, because the biofortified crops cost exactly the same as non-biofortified. Now, on the way forward, we identified 8 key questions that will determine the extent to which biofortification will realize its full potential. I don't have time to talk about all 8. 5 of them are highlighted in yellow. They discuss particular points within biofortification. 3 are highlighted in green. They talk about biofortification as one of several agricultural strategies that can be implemented. I just want to highlight one in yellow and one in green. The one in yellow is tapping into the potential of genetic engineering. I feel that's really key because multiple traits can be introduced into single varieties. But under present regulatory systems, we've seen the potential being really constrained. So the key question is whether those regulations can be lifted to a great extent so that we can really tap into the potential of genetic engineering. Under agricultural strategies, I want to point out the constraint of whether policymakers really do give priority to human nutrition objectives. We've made a lot of strides in the nutrition community now. They recognize the importance of food systems and quality diets and improving mineral and vitamin intakes. And if we're going to work well and closely in the agricultural sector with the nutrition community, the agricultural policymakers really have to be tuned into this and really, give priority to human nutrition objectives. So with that final point in mind, I wanted to present this last slide. I've made several trips to FAO, and during one of my trips, I was standing in the lobby waiting for someone, and I noticed this small plaque on the wall, and I just went over to read it. Let me read what it says. In this building, 16th of October, 1945, so exactly 75 years ago, representatives of 44 nations met and established the Food and Agricultural Organization, first of the new United Nations agencies. Thus, for the first time, nations organized to raise levels of nutrition and to improve production and distribution of food and agricultural products. And I think what really struck me was that they mentioned nutrition first, and they mentioned the production of agricultural products second. So I really like that order of priority, and I was really struck that back in 1945, that was the order of priority. So with that in mind, thank you for listening, and we'll open it up now to questions.
Thank you, everyone, for an excellent overview of the highlights, the key information from the paper. We're receiving several questions that we will address with our panel in just a few minutes. As we prepare, let me share some information about an upcoming event. We have a release of another CAST publication scheduled for November the 17th on ground and aerial robots for agricultural production, opportunities and challenges. And this will be led by Dr. Santos Pila at the University of Nebraska-Lincoln. We invite you to join us for that. And if you're not receiving invitations to CAST, please let us know through our website, and we'll be happy to add you to that list. Once again, let me welcome everybody back and turn the program over to Peter Goldstein, Head of Strategic Communications with HarvestPlus, who is going to serve as the moderator for our Q&A session. Peter, thank you, and we'll turn it over for some question and answer. Okay.
Thank you very much, and thank you everyone for those great presentations. We have several questions, and I think we'll start off, I think this is for Matin, but maybe Howdy or others might want to weigh in. Questions about the economics of biofortification. Specifically, how is it that biofortified crops don't cost farmers any more than non-biofortified crops? If you could elaborate a little bit on that and explain the economics a little bit more. Thank you. Matin?
I can, maybe I can take that one.
Okay.
The basic idea behind that is that we're breeding the minerals and vitamins into high-yielding crops. It may be that in the beginning there's a really strong demand for biofortified crops, and maybe the price of the biofortified crops is higher than non-biofortified. But you'll get a supply response in that case, because it'll be much more profitable to grow biofortified crops. The supply will go up, and in equilibrium, you'll get an equal price because of the high yield. So if you have a basmati rice, say in India, the price is always higher of basmati rice because the yield is lower. So that's the basic reason why the price is the same. So when you substitute the biofortified crop in the diet 1 for 1, you get more minerals and vitamins in the diet for the same price. And so that's a great incentive for the consumption of the biofortified crops.
I mean, perhaps I can add to this. Many people may feel that biofortified crops cannot be reproduced, but they can. Right, so it's not a requirement that farmers have to return to the seed market every year, but they can also reproduce their seeds, and that's something where, of course, which is of course important to ensure widespread adoption and continued adoption, and that's also one of the reasons why the costs for farmers are low. And I think that's one of the reasons also why in the end the cost effectiveness is so high.
Thank you. It's kind of a follow-up to that question in terms of the economics and what we should be investing in. As you pointed out, Matin and others, the main problem is that people are not able to afford diverse, nutritious diets. So shouldn't we really be investing in greater production or availability of fruits, vegetables, meat, and et cetera, instead of investing in more nutrients for staple crops?
I mean, this is not an either-or question. Of course, the long-term objective should not be that people get all of the nutrients from staple food crops. Diverse diets with nutritious fruits and vegetables and pulses, this is the long-term objective. But we are looking here at people who are generally poor and cannot afford more nutritious foods in sufficient quantities. We are looking at seasonality issues. We are looking at knowledge issues. And that means we should not do one thing and leave the other. Of course, we need to make access to nutritious foods better, and that's not an alternative to biofortification, nor is it the other way around. But whereas the nutritious diets for all, diverse and nutritious diets for all is more the longer term goal, I think we can't wait until this longer term goal is achieved.
and certainly need to look for short-term effective interventions, and biofortification is definitely one of those short and medium-term interventions.
Okay, thank you. There's also interest, this is for Dominique, I believe. You mentioned, I think briefly, new genetic breeding, genetic engineering techniques, for example, CRISPR. We just saw recently that those who had been working on that technology just received the Nobel Prize. To what extent can this new technology really drive forward biofortification, particularly in terms of timeline, when it might become something that will have a real impact on the breeding process for biofortification?
Thank you, Peter. Well, I think it's indeed an important and timely question in view of the Nobel Prize that was given for CRISPR. It's a great technology. It has many advantages, but I would not propose it as the golden bullet that will solve all the problems regarding biofortification, certainly not regarding multi-biofortification as it stands today, at least. Well, first of all, I think there are three different aspects that we have to consider. Where can CRISPR definitely help? First is the fact that we can direct a DNA cassette towards a so-called safe harbor location in the genome, meaning a location where basically an insertion of a piece of DNA will not have morphological consequences. The second one is that CRISPR is a great help to excise marker genes, and this actually also has been part of the rejection of GM crops initially because of the antibiotic resistance genes that were used in the beginning as selectable markers. So these markers can be excised precisely using CRISPR technology. And then third is, of course, the fact that for single genes, there is already quite some evidence that indeed using CRISPR, one can confer, for instance, bacterial resistance. And then I will refer to the recent example of Xanthomonas oryzae, that's bacterial blight resistance in rice. But there are also other possibilities, like, for instance, making a CRISPR variant of golden rice, for instance. Now, the point is, of course, can CRISPR help also in multi-biofortification? So as mentioned, first of all, regarding the safe harbor location, for sure, regarding excising the markers, for sure. But the point is, can we raise the densities of multiple micronutrients using CRISPR only? And then I think the answer today is no. And the reason why is, first of all, because for several micronutrients, in order to raise the levels of these micronutrients, we need to tackle several genes. I will give the example of folates, for instance. In order to reach a high density of folates together with stability of the molecules, we need to use at least 3 and optimally even 4 different genes. If we go to the example, the well-known example of golden rice, there we know that 2 genes were used. If we look at the rice which was created by ETH in Zurich with enhanced levels of provitamin A, iron and zinc, there a combination of 4 genes again was needed. And obviously the point is, can we, with CRISPR technology, with, for instance, single nucleotide changes, can we raise the levels of all these different micronutrients at once? So today, as the technology stands today, I think the answer is no. I'm not saying that it won't be possible. Obviously, technology is there also to be refined. I'm sure CRISPR technology will still be refined and already has been refined. We shouldn't forget that. It has been actually launched in 2012. The system was known already for over 30 years in bacteria, but actually used in other organisms since the publications of Jennifer Doudna and Emmanuel Charpentier in 2012. But in the meantime, it also has been made more precise. That is called prime editing. So technology, of course, is evolving. It will definitely still be refined and offer new possibilities. But as it stands now, I think CRISPR is a perfect aid to gene engineering technology, to metabolic engineering technology, in order to reach high densities of different macronutrients and also in a stable form. I hope that was clear.
Yes. Would anybody like to add to that, or should we move on? Okay. Related to that, there were questions, and I'm throwing this out to the group, about acceptability of by countries of genetically engineered crops. As Howdy pointed out, this is not widespread at the moment. I wondered if there's any. The question is, what are the prospects for that expanding in the near future, and what are the dynamics of that? Perhaps Howdy, you would like to respond to that, or?
Well, I'm not really an expert on deregulation and how all that's going, but I do hope that when golden rice is fully deregulated, we're close in the Philippines. That once it's deregulated and it's actually being sold to farmers and it's actively being pursued, I'm hoping there'll be a real sea change in the debate, and people will understand that this is really valuable technology. We've proven with conventional breeding, we've proven that biofortification works. So we've taken that question off the table in some sense. Now we can put multiple nutrients in the same crops. Already we've got high iron and high zinc rices through transgenic methods, much higher than we can do with conventional breeding. So we can combine that with golden rice. We can put iron, zinc, and vitamin A in very high levels in the most widely consumed staple food in the world. And that can have a huge impact. And if people would just realize that, we just have to get past this, these regulations. All the national academies of science around the world have said that transgenics are safe. All the fears, aren't based on the science.
Thank you. We have a question for Ekin regarding, you mentioned the priority of mainstreaming biofortification, basically mainstreaming, targeting nutrient traits. The question is, obviously the CGIAR centers such as CIMMYT and IRRI have enormous impact on breeding programs globally. Can you comment on sort of the level of commitment to mainstreaming, and what is the strategy going forward in terms of expanding that?
Yeah, absolutely. Thank you. Can you hear me?
Yes.
Yeah.
Okay, great. Thank you so much, and thank you for the question. It's an excellent question because it's very important for scaling up of biofortification to become the new normal or go viral, as I previously mentioned. As several of you might know, the CGIAR is going through a reform called 1CG, and it's a great opportunity to use the modernized breeding groups or programs that are being developed to include micronutrient traits in breeding programs. At the moment, or in the past, since HarvestPlus has started, we've been doing targeted breeding. HarvestPlus crop development team, especially our chief scientist and director of R&D, Wolfgang Pfeiffer, working very closely with all the CG centers to implement these targeted breeding programs. But moving forward for mainstreaming, for making sure the biofortification gets integrated in every single variety that will be coming out of the CG, it's very important that all CG breeding centers are committed to including nutrients as some of the core traits. So this is, I don't know if Howdy wants to add anything. And yeah.
I would like to add something.
Oh, yes, please.
Sure.
Thanks.
And not only the CG centers that need to mainstream into their programs, but we always work closely with our national partners. And I think one of the things I'm most proud of in our sweet potato breeding network is that 14 sub-Saharan African country national program committed that at least 50% of whatever they submitted to a release committee would be biofortified with high levels of beta carotene or provitamin A. So it's a joint effort. And I think Ekin also, the recent decision in India, I think, where they set the standards that now any pro-millet variety that's going to be released has to have a certain level of iron to be considered. So I think really it's together working with national programs to really promote and expect that these varieties are going to become the norm as part of the strategy as well.
Thank you. We have a question. I believe this one would go, this is more about the nutritional aspect of the crops, either to Saurabh or Torbert. And the question is about intrinsic absorption inhibitors in the biofortified crops. What factor does that play in the development of the crops, and what challenge does that pose?
I may not be the best person to answer this. You know, there's been work on phytic acid and that's effects on absorbing iron, and that's an area of work that's been done. It's somewhat controversial because there have been associated effects with yield, but I'm pretty much focused on provitamin A, and there is variation for bioaccessibility and bioavailability in the different crops, and that could be looked at with newer technologies in a relatively straightforward way. It's something to think about, but I also just think breeding for more. We could breed for more of these micronutrients, and we know what they are, and it's more straightforward than assessing bioavailability and bioaccessibility, because that's very complex. And when a breeder makes a cross and they go into another line, it's going to be hard to follow that. I'm not saying don't do it, and we should do it, but so I've kind of given you there's different aspects of it, and maybe somebody else could comment on this as well.
So Rob might want to make a few remarks on this. It's always, you know, what will be the impact from a human nutrition point of view, and then what's possible from a breeding point of view. So Torbert addressed it from a breeding point of view, so Rob might want to say some remarks from human nutrition.
Absolutely. And as Torbert already pointed out, I think it's a really complex topic because definitely there are phytates and polyphenols that might be inhibiting the absorption of certain nutrients like iron, but reducing them may not necessarily be the answer because they also have their own health effects, such as phytic acid has been shown to attenuate iron-induced lipid peroxidation that might have its own effects on kidney stone risk and osteoporosis risk. There is some research which is again in the pipeline that is looking at promoter substances. So how do we add promoter substances in some of these crops, such as like nicotinamide, which is a non-protein amino acid, and it has been shown to both promote iron bioavailability in crops as well as particularly crops that contain high levels of phytate. So that might be one way to go, but just reducing phytates or polyphenols may not be the answer.
So I mean, I think it's an area that merits research, and we will learn some things, but it's clearly more complex, and there's interactions we don't know about, because you have iron, you have zinc, and you have provitamin A, and then there's a whole array of other compounds that may be promoters or inhibitors. So I guess I would say we need to work on this, and we need to do it steadily in conjunction with the straight breeding and transgenic efforts for the actual micronutrient.
Thank you.
If I could just add
Yeah,
something,
sure.
Peter, to this discussion, because been collaborating with ETH Zurich on looking at bioavailability of high iron potato, and we had an enhanced iron variety of sweet potato that we did bioavailability studies on. And the results in the potato were excellent. We were very pleased with the level of bioavailability at over 30%, 28 to 30%. But the sweet potato, the high iron sweet potato, the iron did not have the bioavailability that we would like to see. And they suspect that it's probably due to the presence of very high levels of polyphenols. Now, polyphenols can be a very positive thing for nutrition. So it is, I think Torbert's right, it's a complex issue, but we must look at bioavailability because that influences the level of iron that we have to breed for, either conventionally or eventually with genetic engineering tools.
Yeah, and Jan, to follow up on that, this makes me think back to one of the very first HarvestPlus crop meetings. It was maize, in where were we? We were, I don't know, Kenya or Ethiopia, maybe, Howdy.
We were in Addis. Yeah, we were in Addis.
Yeah, and so we really didn't know what we were doing then. We hadn't even started. You know, it was a month after the funding was approved, or 2 months. But I do remember Kevin Pixley, who was the maize crop leader, he was just putting up on the board vitamin A, iron, zinc, what are we going to work on? And then there were target levels and what's kind of out there for natural variation. And then there were certain assumptions for bioavailability. And then we were just looking at the easel and we were like, well, gosh, if we just increase bioavailability a little bit, it would go a long ways in hitting target levels. So there is real potential there. Just speaking from myself, I work on carotenoids. There's a well-known pathway. People have been working on carotenoids, doing biochemistry for 70 plus years, and we know about it and we've made progress with that. The bioavailability is more complex. Somebody else can work on it. And I don't mean that in a bad way. It was like we made progress with a biosynthetic pathway, and now we have a lot more knowledge. So maybe someone else can make more progress with bioavailability.
Thank you, Torbert. Howdy, we're close to the top of the hour. Do we have time for more questions, or would you like to wrap up?
It's up to Kent, really.
I think we
Okay.
probably need to wrap up. We got about 2 minutes left, so.
Okay, thank you everyone for answering the questions.
Well, I'd like to add my thanks to the panel and also their work as authors in helping put this paper together. If you've not accessed a copy of the paper, it is available on our CAST website, www.castscience, under our publication tab. It is a free download, and we encourage you to go there and download it, as well as the quick cast summary and the student study guides that are there as well. And then also here's an email for contacts for our panelists today if there's any particular questions. We will gather some of the questions we weren't able to get to, send those out to the panelists, create kind of a question and answer document that we'll post to our CAST website or circulate at a later time. Again, thank you to our panelists. Thank you for the great presentation and all the work that went into doing this paper. We appreciate the opportunity to